Good morning. It's March 30th, and this is your daily brief in science. Here's everything you need to know. A new RNA-based therapy aims to turn the body into its own drug producer to repair the heart after a heart attack. Researchers at Columbia University report that this approach boosts production of ATRIAL natriuretic peptide, or ANP, by delivering an inactive precursor that becomes ANP in the heart to promote repair and reduce scar tissue. This is a multi-department effort at Columbia moving toward a Phase 1 safety trial at Columbia University Irving Medical Center, with in-house manufacturing support. The therapy uses self-amplifying RNA in a two-step mechanism, aiming for effects that last up to a month from a single injection and to reduce the need for invasive procedures. Preclinical results in mice and large animals show reduced scar tissue, improved heart function, and effectiveness even when administered days after a heart attack, including models with diabetes or atherosclerosis. In studies, a single intramuscular injection reduced scar tissue and improved heart function across small and large animal models, with effects lasting at least 4 weeks. The approach could minimize side effects by targeting activation to the heart and may lower treatment barriers by reducing hospital visits. If successful in humans, this non-invasive RNA delivery could transform cardiovascular care and potentially extend to other conditions like kidney disease, high blood pressure, and preeclampsia, with human trials planned at Columbia University Irving Medical Center. If successful, the non-invasive RNA delivery could offer a simpler alternative to heart transplants or cell-based therapies and might be adaptable to other organs and conditions such as kidney disease, hypertension, and preeclampsia. Researchers demonstrated the strategy with the NPPA gene, the precursor to ANP, and showed delayed treatment at 1 week post-heart attack remained effective across models, including older age, atherosclerosis, and diabetes analogs. Funding comes from the American Heart Association and NIH. The study was published in Science on March 5, 2026, under the title Single Intramuscular Injection of Self-Amplifying RNA of NPPA to Treat Myocardial Infarction. A new study presents the earliest direct evidence that plate tectonics was active on Earth about 3.5 billion years ago, reshaping our view of early Earth and life. Paleomagnetic data from the Barberton Greenstone Belt in South Africa show near-stationary behavior for that period, supporting a model of a lithosphere segmented into moving blocks rather than a single global shell. Findings from the Pilbara region reveal a latitudinal shift from 53 degrees to 77 degrees and a clockwise rotation exceeding 90 degrees over tens of millions of years, indicating significant early plate motion. The study, led by Harvard University's Roger Fu and published in Science on March 19, includes corroboration from Yale and other researchers. The researchers argue against a uniformly moving single crust and instead support a segmented early plate model with relative motion between blocks. The conclusions combine new paleomagnetic data with reanalysis of existing records, contributing to a revised narrative of Earth's early geologic history and its climate and habitats. Independent scientist Uwe Kircher emphasizes the abundance of paleomagnetic data from ancient rocks and its importance for understanding relative plate motion in Earth's transition to plate tectonics. Context on today's plate dynamics notes that Earth currently has 7 major and 8 minor plates that move at a few centimeters per year, with most activity at plate margins. Lead author Alec Brenner and the team conclude that Earth's lithosphere was already divided into multiple movable segments 3.5 billion years ago, signaling an early move toward modern plate tectonics. The research is based on 900 rock samples from the East Pilbara Craton in Western Australia, using paleomagnetism to infer ancient latitudes and plate motion through magnetic orientations in minerals. By establishing an early start to plate tectonics, the study helps explain the formation of Earth's mountains, oceans, climate, and the habitats that supported early microbial life. Ultimately, this work reframes how we understand Earth's early environment and the conditions that allowed life to thrive. A GPU-accelerated approach from Arizona State University is mapping slow vibrational modes that drive protein shape changes, offering a roadmap to explore relevant conformations in under a day. Associate Professor Matthias Hayden and team capture slow, low-frequency motions from nanosecond simulations, making long time-scale transitions observable. The method moves beyond static structures, building dynamic ensembles that could support a sequence-to-structure-to-dynamics framework for predictive proteomics, alongside advances like AlphaFold. Published in Science Advances on March 27, 2026, the work shows proteins sampling multiple conformations by analyzing thermally driven fluctuations at room temperature with robust, repeatable results. Practically, this accelerates drug discovery for targets in antibiotic resistance and cancer by enabling high-throughput exploration of conformational landscapes with accuracy. Dynamic sampling enhances understanding of allosteric effects and can reveal subtle binding sites and how ligand binding propagates structural changes. Applications span protein engineering and synthetic biology, including the potential to create smart proteins with switchable functions and improved catalytic efficiency through controlled dynamics. The workflow uses ASU's Sol supercomputer and GPU parallelism to democratize access to dynamic protein simulations, making routine exploration feasible for labs worldwide. This GPU-accelerated method enables observation of conformational transitions that were previously time-prohibitive, broadening the practical scope of molecular dynamics studies. Future prospects include integrating with experimental techniques like cryo-EM and NMR to build richer dynamic portraits and refine predictive models of protein motion. A decade-long international effort has mapped the Antarctic Southern Ocean microbiome to understand its role in Earth's climate, revealing substantial heat and carbon uptake and the critical importance of microbial processes. The study argues that decoding the genes controlling microbial functions is essential for predicting how the Southern Ocean will influence future climate, and it outlines plans to explore newly discovered genetic diversity. Microbial communities there are not uniform. They form distinct ecosystems shaped by ocean circulation, with some organisms thriving in cold surface waters and others at greater depths. DNA from water samples collected in 2016 to 2017 was sequenced and compared with existing gene catalogs, showing that at least one-third of identified genes are not in current marine databases, indicating largely uncharacterized genetic territory. The findings were published in Nature Communications on March 9, 2026, in a paper titled Water Mass. Specific genes dominate the Southern Ocean microbiome, led by Nicholas Kasser of Duke University as part of an international team. Phytoplankton, a key microbial group, drives roughly half of the planet's photosynthesis and plays a major role in removing CO2 from the atmosphere, underscoring their central place in the carbon cycle. The research highlights that understanding gene function in these communities is needed to predict how heat and carbon uptake in the Southern Ocean will evolve with climate change. It details a plan to investigate the newly discovered genetic diversity to better model future climate responses. The study emphasizes that the Southern Ocean's microbial activity is a significant factor in global carbon cycling and climate projections. Overall, the work positions the Southern Ocean microbiome as a crucial frontier for understanding how microbial processes influence the planet's climate system. A single study maps the epigenetic landscape and gene regulatory network of memory T cells across tens of thousands of human cells from multiple donors. The work uses comprehensive single-cell genomics and regulatory modeling to show a core set of 5 transcription factors: KLF6, MAF, PRDM1, RUNX2, and SMAD3 that maintain readiness and drive rapid responses when antigens are encountered. The findings have implications for next-generation vaccines, especially for the elderly, and for precision therapies that curb pathological immune activation without broad immunosuppression. Memory T cells retain a pre-open chromatin architecture at many immune-related enhancers and promoters, enabling faster transcriptional activation upon reactivation. Integration with genetic data from over 100 individuals links memory-specific regulatory elements to DNA variants associated with asthma, allergies, and autoimmune diseases, suggesting modulation of immune activation rather than changes in protein-coding genes. The work was published on March 26, 2026 in Cell Reports, with full citation and DO1 provided. This collaborative interdisciplinary effort is supported by NIH funding and underscores the resource-intensive nature of the study. The research presents a systems biology view, portraying immune memory as an emergent property of interconnected transcriptional networks and epigenetic states rather than isolated gene effects. Memory CD4-positive T cells are genomically primed to respond rapidly upon pathogen reencounter, in contrast to naive T cells that require chromatin remodeling. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.